Hello and good afternoon friends, welcome once again to the CEC-UGC live lecture. Dear friends, as you know that yesterday we discussed about bio aerosols. We discussed what is bio aerosol, what are its types, as well as numerous things we try to understand with the previous lecture. And today also we are going to continue with the same topic, that is bio aerosols, and we would be discussing something new under this very topic. And for this, again with us in our studios we have Dr. Arun Kumar Srivastav. He is Assistant Professor in School of Environmental Sciences, Jawaharlal Nehru University. So first of all, I would like to welcome our guest, Dr. Arun Srivastav. Hello sir, welcome to the EduSet lecture. As in previous lecture we discussed about what basically bioaerosols are and what are its types. What basically are we going to study today?
Well, in yesterday's class we discussed about the definition, etc., the types of bioaerosol, what exactly bioaerosols are, and the next important thing in that was how it can be sampled, how it can be collected, because bioaerosols are present everywhere in any kind of environment, whether it is humid, arid, or with low temperature, high temperature, with the presence of sunlight or with the absence of sunlight. So bioaerosols are ubiquitous. But the thing is that it is very difficult, it is not very easy to collect them. How they can be properly collected, how they can be properly sampled, then only the next step, that is this analytical parts, starts. Without sampling, analytical or understanding the bioaerosol, its origin, its pathogenicity, its other characterization will not be possible. Thus, the sampling is a very important part, and we have already discussed it in the previous lecture, what different kinds of sampling methods are, how they can be sampled, as well as this sampling techniques are divided on different bases, on the basis of its cost, on the basis of convenience, like its occupancy of the space, how much space it can cover, and in fact in different regions or in different conditions how it can be applied or where exactly the need of that particular type of samples are. So in the previous lecture we discussed about those things, and now we will discuss mainly about its characterization, how the bioaerosols, the collected bioaerosols can be characterized or can be assessed. There are two things. One is its concentration, how much the amount of bioaerosols are present in any particular environment. So that is one of the things. So in this particular lecture we will discuss about that how bioaerosols can be assessed as far as its concentration is concerned. Apart from that, we will also try to understand how it can be characterized. Characterization means what exactly type of that bioaerosol is, whether they are pathogenic, harmful, harmless, allergic, etc., etc. So let us start with it. So as far as assessment methods of bioaerosols are concerned, they have broadly been divided in two categories. One is culture based method, another is non-culture based method. Now, what is culture based method actually? In this method, this method is mainly and precisely applicable for bacteria and fungi, because in the previous class we have discussed the bioaerosols are of different kind. They could be viruses, they could be bacteria, they could be fungi, they could be protozoa, they could be algae, they could be molds, etc., etc. They could be living, they could be non-living. So in culture based method, only those bioaerosols are covered which may have the capacity to growth or have the capacity to further multiply. So in this particular case, the culture based method, the bioaerosols especially of bacteria and fungi types are covered. Now, how this culture based method is done, what are the different steps of a culture based method? That is, now the first one is the culture and growth, the next one is sample preparation, and the third one is enumeration, and the fourth and that is the last one is identification. So what do we do exactly? In the culture based first step of the culture based method, we try to grow the bacteria or fungi which we have collected, and once it is sufficiently grown or it is grown in enough quantity, afterwards we prepare the sample. I mean, a very small portion is taken out of it and it is placed on a when it is grown completely, then sample is prepared, and then the next step is enumeration, and we will in fact discuss one by one properly, and the last one is identification. Now, first one, that is culture and growth. What is done in culture and growth? After sample collection on a specific media, colonies of bacteria and fungi are grown on culture media at a definite temperature over a fixed duration. So, here in this particular table, different temperature as well as the duration for sufficient growth is given. Like in case of bacteria, the media which is required for its optimum growth is tryptic soya agar, that is also called as TSA, casein soya peptone agar, that is CSPA, or nutrient agar, and the temperature, the required temperature for its optimum growth is between 30 to 37 degrees centigrade, and the duration for its complete growth is between 1 to 5 days. This variation is basically because of taking into account the different type of bacteria. Some bacteria can grow at a very comparatively at a lower temperature, like at around 30 degrees centigrade, at even in lesser days than the other end of the 1 to 5 days, that is, it can grow in 1 or 2 days. While in case of fungi, the media which is used are malt extract agar, that is known as MEA, as well as rose Bengal agar, that is RBA, and the optimum temperature for its growth is 28 degrees centigrade, while the duration for its growth is 3 days. Now, in this particular picture, it is beautifully shown here how the growth is taking place. In the first slide, we will see very few colonies or very few amount of bacteria are there, just scattered at one, or it is in the form of spots, 1 or 2 spots, while in the second one we will see that the bacteria are grown in some amount. I mean, we can efficiently see that some amount of bacteria or visible colonies are there, while in the third one a sufficient growth has taken place, and the bacteria is more or less completely grown. Now, the next step is enumeration. How the enumeration is done? Once the bacteria or sufficient colonies are grown, then our next step is to count it, and this can efficiently be done with the help of a colony counter. Once the colony is counted, that is also known as colony forming units per cubic meter, and the concentration of the bacteria can be determined in 2 different ways. That is, one is total concentration, that is CFU per meter cube, or adjusted concentration, that is again the unit of that is in CFU per meter cube. Now, what is total concentration? In fact, the total concentration of a culturable microorganism is calculated by dividing the volume of air sampled into the total number of colonies observed on the plate. So, what is done in this actually? The total number of colonies are counted first, let it be n number of colonies, and then our aim is to divide it by the total volume of air that has been passed through during the entire sampling period. Once this is done, so we can further recalculate it in the units of volume of the air, that is, the finally will get CFU per meter cube, or the concentration of colonies in a cubic meter of ambient air. Now, here it is also given a colony is a microscopically visible growth of microorganisms on a solid culture medium. CFU is the number of microorganisms that can replicate to form colonies as determined by the number of colonies that develop. Now, what is adjusted concentration? In fact, often it is difficult to identify multiple colonies at one location on a plate because of the lack of differential colony morphology, or because the chemicals secreted by one microorganism might inhibit the growth of other organisms at that same location. So, the adjusted concentration of a culturable microorganism is calculated by dividing the volume of air sampled into the adjusted number of colonies observed on the plate. Because this the idea of this is that what happens, sometimes the colonies overlap. So, it is very difficult to count them, or the chemicals which the bacteria or fungi secrete, it can inhibit the growth of the growth of some of the bacteria. So, the overall information about the total number of colonies we get is usually hazy, it is not clear. So, just to overcome that, we have applied, or we adopt a different kind of method, that is known as adjusted concentration of culturable microorganisms. In fact, this is the best representation of the concentration of microorganisms or the bioaerosols once it is divided by the volume of the air which is being sampled. Now, the next step is identification. What is done in identification? In fact, identifications are of two different types. One is microscopic identification, another is microbiological identification. Now, what is done in microscopic identification? In fact, once the colonies are formed onto the Petri plates, a small portion of that particular colony is taken with the help of inoculum loop and placed onto a slide containing 4% of NaCl or sodium chloride, and then a drop of lactophenol cotton stain is added over it and left for about 1 to 2 minutes. Then the area is covered by a cover slip and is ready for microscopic examination. Now, in case of microbiological identification, pure culture of any bacteria or fungus is done, and from the pure culture DNA is isolated. With the help of PCR, magnification is done, and then with the help of gel electrophoresis, visualization is done, and afterwards the next step is sequencing. And once the sequencing is done, and then it is not very difficult to identify what kind of species is that. I mean, with the help of this, we can identify that particular bacteria at a species level. Now, in non-culture method, this method is basically applied for non-viable bioaerosols such as pollen, etc., or mycotoxins or endotoxins. Now, non-culture method is of 5 different types. They are direct examination, staining, microscopy, gas chromatography, spectrophotometry. Now, what is done in direct examination? In direct examination, a hemocytometer chamber permits the determination of total number of bacteria. This technique, in fact, can be applied as well when information about the arrangements or other distinctive morphological feature is needed. Non-viable bioaerosols, especially pollen and spores, can best be visualized by this method. In staining, bacterial membranes, cell walls and capsules can be stained with a specific dye to reveal valuable information about the microbial content of sample, such as bacteria impacted upon glass slide or in liquid. Now, I think in the continuation to that, in microscopy, one is light microscopy. In this particular case, many bacteria, pollen and spores still rely on bright field light microscopy. However, this method cannot give much details of bioaerosols except general morphology, while in case of electron microscope, with the help of electron microscope, the shape and other morphological characters such as branching, filaments, pointed end or granular forms, etc. I mean, with the help of electron microscope, we have a broad idea of its morphological characteristics. Now, here are given here some pictures about the airborne common bacteria which have been identified and pictured with the help of a microscope is given, and in fact, we can see in this particular picture there are 6 different types of, I mean there are more, but here 6 different type of bacteria are provided. They are Alternaria, Aspergillus, Basidiospores, Curvularia, Cladosporium, Buteratis. And here also we can see some again common airborne bacteria which are usually found in the air, and they are Penicillium, Epicoccum, Rhizopus, Ulocladium, Fusarium and Mucor. These have been done with the help of a microscopy attached with a camera. Now, the next tool is gas chromatography. What is done with the help of gas chromatography. In fact, with the help of these type of tools, mycotoxins are detected. We will discuss more. We will discuss elaborately about mycotoxin in coming slides. But first, we will have some information about mycotoxins. In fact, mycotoxins are harmful complex organic compound secreted by a bacteria. Bio aerosols collected filters are sonicated. How these mycotoxins are determined with the help of gas chromatography is given into this. First, what do we do? Bio aerosols collected filters. I mean, this can be done only when the bio aerosols are not collected onto a onto a media, but onto filter papers. Once they are collected on filter papers, then those filters are put in sonication unit, and there it is sonicated with the help of suitable organic solvents, and then it is extracted. I mean, the mycotoxins, which are mainly the organic compounds, they are extracted, and afterwards these extracted samples with the help of liquid chromatography or liquid chromatography may be used for quantification of mycotoxins. I mean, there is a mistake, this is not gas chromatography, but liquid chromatography. And the next is spectrophotometry. This is also a tool for the detection and quantification of bio aerosols. In this type of, with the help of this type of tools, we can determine and detect the endotoxins. Endotoxins are harmful complex organic compounds secreted by bacteria only. Bio aerosols collected filters with the help of water is extracted in a suitable tube, and afterwards these extracted samples with the help of suitable assay kits is placed in a spectrophotometer for the quantification of mycotoxins. Now, what are the different sources of bio aerosols? In fact, bio aerosols are present everywhere. Anything either can be a source or it can be its reservoir. If it does not act as a source, it can act as a reservoir, and even if it does not act as a reservoir, then it can act as a decementer. So, what are the potential sources of bio aerosols? They are generally all the forms of earthly things like plants, soil, water, animal. It can act as a potential source of bio aerosols. In case of animal, it includes human also. Now, what are the practical sources of bio aerosols? They are of different 4 types. One is waste disposal facility, agriculture, food production and other areas. In fact, here all these are very good source of bio aerosols, but their type of bacteria, fungi or viruses etc. emitted could be of very different type and different nature. Now, starting from waste disposal facility. In fact, waste disposal facility of this type of class is again subdivided into different 5 types. They are biological waste treatment facilities, residual waste treatment facilities, materials recovery and transfer stations, landfills, wastewater treatment plants. All these are big and very important source of bio aerosols. In fact, from these places these are the sources where we usually we can expect harmful or very injurious type of bio aerosols. although those bioaerosols which are emitted from agriculture or food production units, if they are not that harmful, but the bioaerosols emitted from waste disposal facility or different type of waste disposal facilities are generally very harmful and injurious to humankind. Now, apart from these, there is another type of source that is known as occupational source of bioaerosols. They are of different 5 types. They could be laboratory, hospitals, industrial workplace, animal houses, residential environment. So, these are the sources where any particular type or particular types of bioaerosol can be generated and can sustain there forever and ever. So, these are the places where a specific type of bioaerosols one can expect, and that is why persons living over there continuously, they are much prone to be exposed to such type of bioaerosols. Now, apart from these, other areas are also there where which can act as a potential source of bioaerosols. They are paper and wood processing units, horticulture facilities, refrigeration plants, and from in that source category we should mention 2 more things. In fact, these sources are have 2 different branches too. They are amplifiers and disseminators. What is amplifier? Amplifiers are basically places, in context to bioaerosols, amplifiers are places where microorganisms multiply or proliferate. Mostly reservoirs are potential amplifiers, while disseminators are devices causing microbes to enter airborne state or be aerosolized, often the reservoir or amplifier. So, what is meant by this is that once the bioaerosols are emitted from any source, although they are in a very small quantity, but once they are in touch or in contact with a suitable or optimum place which have the potential to amplify it, then it can be a potential source or indirect important source too. And such places are humid places, like they are humid areas in a house, or waterlogging or water bodies in the vicinity of localities. So, these are the places where the amplification of the bioaerosols can take place, and sometimes the number of bioaerosols become huge. Significantly, these amplifiers increases the quantity of bioaerosols. And as far as disseminators are concerned, these are the devices where the microbes can enter into the atmosphere or disseminate into the atmosphere. Often these things are associated with the reservoirs or amplifiers. So, in reservoirs or amplifiers we also find disseminators. So, we can say sometimes at one place we have source, we have amplifiers, and we have disseminator. If such type of situation is there, so that is a very important and very significant source of bioaerosol. Now, what are the different health effects of bioaerosol? So far we discussed about the sources of bioaerosols and how it can be characterized. Now, we should discuss, or we must have some idea about the health effects of bioaerosols. Bioaerosols can cause 2 different type of health effects. One is general health effect, another is a specific health effect. What is general health effect? Bioaerosols actually induces into human body either by inhalation or by deposition on wounds. A possible reason of sick building syndrome is the presence of bioaerosol in the building. So, you will be surprised what is exactly this sick building syndrome is. I think we have already discussed this sick building syndrome in previous lectures, that sick building syndrome is a kind of condition in which what happens once we enter into a building. Before entering we are absolutely normal, we do not feel any problem, or we are not suffering with any kind of problem, but once we enter into that particular building we start having some suffocating effect or some problematic effect. That is known as sick building syndrome, and once we are out of that building, this effect or this type of condition disappears. This is called as sick building syndrome, and this type of buildings are mainly the potential sources of Bio aerosols, or it happens where the accumulation of Bio aerosols are significant, because some people are very much prone or very much vulnerable to Bio aerosols. So, those people, when they enter into a building, they are heavily or they are significantly affected by such type of Bio aerosols, and that is why they always feel problem while entering into such kind of building, and this is why it is known as sick building syndrome. This type of syndrome are generally not for everyone into that particular building, but it can be for some people who are especially much vulnerable to any particular type of Bio aerosols. Now, the next is, despite the defense mechanism of the body, these Bio aerosols could cause damage to the body. The infections caused due to Bio aerosols there are so many, or there are a spectrum of diseases, but here we will discuss few of them. They are Legionnaires disease, humidifier fever, cold, influenza. These are the common or general health effect, and as far as the specific health effects are concerned, they have broadly been subdivided into three categories. One is infection category, another is allergy, and the third one is poisoning or toxicity. What is infection category? In fact, in infection category, infections generally result from multiplication and growth of microbes inside human body. In infection, the Bio aerosols, especially the bacteria or fungi or viruses, enter into our body through the medium air, and once it is entered into our body, it starts multiplying, or it has the capacity to overgrow inside our body. So, once it is overgrown, it overpowers our body or our other immune system, which can come into form of any disease, maybe it is a fever or any kind of headache or stomach, etc., etc. Apart from this infection, the next specific health type is allergy. Allergies are the result of once one is exposed to antigens, and poisoning or toxicity basically result from the contact with microbial toxins such as endotoxins or mycotoxins. Some of the Bio aerosols, if they do not have the capacity, or even if they are dead, they cannot overgrow or they cannot multiply once they are entered into our body. But what happens, they have the potentiality to have or contain some toxic elements such as mycotoxins or endotoxins. So, these mycotoxins or endotoxins, they are very injurious and very harmful, or in other words we can say they are toxic to our body. So, once they are enter into our body, they can cause many type of diseases. Sometimes they are short term diseases, sometimes they are long term diseases, and even in some cases they are also said to be carcinogenic or mutagenic too. Now, starting from infections, infections can be caused by viruses, bacteria, fungi and molds, because these have the capacity to overgrow or multiply. Now, here in this particular table, some of the diseases which can be caused by viruses are given. These are basically airborne. These can be caused by Bio aerosols. So, starting from this is first is common cold, and the virus responsible for such type of problem is rhinovirus, and the size of such type of viruses are usually 0.02 to 0.3 micrometer, and the medium is basically human body acts as a source, but medium for its travel is air only. So, that is why it is either it in itself is a Bio aerosol, or it is deposited on some kind of aerosols. The next one is influenza. Influenza is caused by influenza virus, and the size of such type of viruses are 0.08 to 0.12, and again the source is human body, while the medium of its travel or medium of its passage is air. The next one is congenital and neurological infections, and the virus responsible for such type of diseases are lymphocytic virus, and the size is 0.02 to 0.3 micron. And the next one is gastrointestinal and upper respiratory infection. It is a very common, or usually we can observe, we can see such type of problems, especially in the metropolitan areas, and the virus responsible for such type of diseases are Coxsackie virus, and the size is 0.2 to 0.3 micrometer. And the next one is a smallpox, and the virus responsible for this is variola virus, and the size is 0.2 to 0.3 micron. The next one is chickenpox and shingles virus, shingles disease, and the virus responsible for this is herpes virida HH3 virus, and the size is 0.01 to 2. And there are other diseases caused by aerosols having viruses are like measles, mumps, and rubella. Apart from that, there is one more important, and nowadays recently it was in news very much, and that was because of the SARS. SARS is the disease which stands for severe acute respiratory syndrome, and the virus responsible for such type of diseases are SARS coronavirus, and the size of such virus is 0.08 to 0.16 micrometer, and the medium is air and human. The next class is bacteria, and in bacteria the major diseases that can be caused are UTI or urinary tract infection, diarrhea, dysentery, and the virus responsible for this is of the size of 1.1 to 1.5 micron, and here it can be multiplied by 2 to 6. I mean, it depends upon the type of virus. There are different strains of such viruses. Apart from that, the next diseases which can be caused by bacteria, airborne bacteria, are food poisoning, toxic shocks, pneumonia, meningitis, etc., etc. Apart from that, cardiovascular infections, food infection, otis media. Again, it is a disease which is caused by a bacteria that is known as Staphylococcus, and the size is 0.5 to 1.5 micron. Then the next one is Legionnaires disease or Pontiac fever, and afterwards anthrax. It was also very much popular recently, and pneumonic plague, tuberculosis, whooping cough, etc. These are the diseases that can be caused by airborne bacteria, and it comes into the category of infection. Next one is fungi. Fungi can cause many diseases. They are humidifier fever, allergic reactions, irritation to respiratory tract, aspergillus and aspergilloma, lung infections, pulmonary diseases, flu-like illnesses. However, the diseases caused by fungi are not long-term diseases, long-term or chronic problem, although they are short-term, but they are much common nowadays. And these are the diseases caused by airborne fungi having the capacity of infecting. The next one is molds. The diseases which can be caused by molds are hypersensitivity pneumonitis and candidiasis. Apart from that, the aerosols in the category of specific diseases of aerosols, the next category is allergies. The allergies can be caused at nose, throat, sinuses, and at lungs. Some of the examples of such diseases are allergic asthma, allergic rhinitis, hypersensitive pneumonitis, that is HP, extrinsic allergic alveolitis, EEA, and formous lung. Formous lung is a kind of disease which is a kind of allergic disease caused by aerosols. And the next category of this specific diseases caused by aerosols are poisoning and toxicity. In fact, this poisoning and toxicity are generally caused by two types of toxins which are found in bacteria and fungi. They are endotoxins and mycotoxins. Now, I will discuss them one by one. What is endotoxin? Endotoxin is basically composed of lipopolysaccharides, that is abbreviated as LPS, and is a non-allergenic cell wall component of usually gram-negative bacteria with strong pro-inflammatory properties. It is commonly present in many occupational environments, but also in the general environments and particularly in house dust. And here there is a list of the diseases which can be caused by endotoxins, bacterial endotoxins, and I said that it is mainly a kind of a toxin which is found in occupational environment. So, we are giving here some of the occupational environments where endotoxins can be observed. So, it is found in cotton industries, and the health effects which can be caused by that particular endotoxins are acute and chronic respiratory problems, and it includes asthma too. Apart from in farming occupational environments where livestock and pig and poultry works are going on, and animal feed and grain industry, and there we can observe or we can expect the health effects as acute and chronic decline in lung function, that is mainly caused by endotoxins in occupational environment. Apart from that, potato processing industry, fiberglass industry, they also do contain some endotoxins where the diseases such as non-allergic, neutrophilic airway inflammation is expected. Apart from that, in slaughterhouses too, endotoxins are expected, where the problem that one can expect is increased bronchial hyperresponsiveness. In waste and compost industry, again we can find some endotoxins where the disease or the problem that is caused is organic dust toxic syndrome, and it is a kind of acute and short-lived flu-like symptoms with fever and airway symptoms. Apart from these, we can also expect some endotoxins in vegetable fiber productions where the chronic bronchitis problems are generally anticipated. Now, what are mycotoxins? Mycotoxins are basically found in fungal excreta. Mycotoxins or fungal toxins are low molecular weight biomolecules produced by fungi that are toxic to both animals as well as humans. Some of them like aflatoxin and aspergillus can be they are said to be, or it has been established that they are carcinogenic. Apart from that, mycotoxins of Fusarium, Aspergillus and Penicillium genera are known to be present in the inhalable fraction of airborne corn dust, cotton dust and grain dust. Now, what could be the health effect of mycotoxins? Still so much has to be studied about mycotoxins, but some of the informations which are available as far as its health effects are concerned, they are immune suppression, rash, headache, fatigue, sore throat, pulmonary hemorrhage, and this pulmonary hemorrhage is mostly confined to the infants, and memory loss. Now, this as far as its health effects are concerned, this Bio Aerosols have got historical purview too. In 2003, April 2003, there was a worldwide outbreak of epidemic SARS, and in fact entire world was concerned about this SARS virus. So, we can see here in this map more or less a good portion of world was covered with such viruses or such type of disease. Entire America and China, they were heavily infected with such viruses, while this virus has its traces to other countries such as India, European countries, South African countries, especially South Africa, South America, Australian countries as well as Tasmanian countries. So, more or less the entire world was covered and concerned with SARS virus and epidemic caused by SARS virus. Now, how we can overcome with this problem or these type of bioaerosols? There are some methods. In fact, these things are still in developing phase, but there are some some remedies are available. So, how this bioaerosols can be separated and purified? There are different type of purification methods. There are sedimentation and flotation. Here we can remove or we can separate primary parasites. The next one is precipitation method. It can be used for the separation of viruses. Filtration method. In this type of system, with the help of these type of systems, all classes of bioaerosols can be separated. Immunomagnetic separations, here again all classes of bioaerosols can be separated. The next one is flow cytometry, where with the help of this bacteria and parasites are separated. Now, in case of secondary concentration and purification, these are the methods and these are the tools with the help of which the bioaerosols can be purified or can be removed. Now, we will discuss some of them, not all, but few of them, like chemical precipitation method. What is done in this? With the help of this, viruses and parasites are removed. So, viruses, what is done? It precipitate with the polyethylene glycol or aluminum hydroxide. What is done? Resuspended polyethylene glycol precipitate in aqueous buffer, and then it is dissolved with aluminum flock in dilute acid solution. Afterwards, both have seen used as second step concentration and purification method. Similarly, the parasites also dissolve and precipitate with the help of dilute sulfuric acid. Now, there are other recovery and concentration methods. Here, we can utilize minerals, synthetic resins and other granular media. In case of minerals, iron oxides and talcs are used to absorb the viruses. In case of synthetic resins, ion exchange and adsorbents are used, while in case of granular media, glass beads and sands are used. Now, how it can be recovered with the help of recovery and concentration of pathogens? There are two different type of methods. One is flotation, another is buoyant or isopanic method. So, what is done in flotation methods? Microbes are basically kept in a medium of higher density than that of microbes, and then we centrifuge it, and what happens afterwards? Microbes start floating onto the surface of that particular medium, and from there this microbes which is now been present at the top layer is removed. And in case of isopanic method or buoyant, in this particular a density gradient is created, where microbes are kept in a medium with varying densities with wet depth, but there is one layer where the density of that particular medium is exactly equal to the density of that particular medium. What's happened after getting such type of density gradients? Microbes start migrating from higher to the liquid where the density is equal to that of microbe, and there it starts accumulating, and from there it can easily be removed. So, now with the help of this we end this particular lecture.
With this note, thank you, sir. Thank you so very much for explaining us in detail. As you explained about, though it is very difficult to collect the bioaerosols, but still I want to know that is there any simplest of the techniques which you have described in the lecture itself to collect the bioaerosols?
Yes, in fact, bioaerosols, collecting bioaerosols, removal and its separation is not very easy, but its collection from the air of bioaerosols are not very difficult. They are established, and there are many methods for the collection of bioaerosols. There are various samplers with the help of which bioaerosols can easily be collected, but we have to, it is not that once it is collected, all the different components of bioaerosols are segregated or identified. What is done? In fact, first we will have to aim what type of bioaerosols we are going to collect. If they are bacteria, so we will have to concentrate on bacteria only. So, what we have to do? We have to prepare a medium which is suitable for the collection of bacteria only. Then in fact, even in bacteria, bacteria are also of two types, one is gram negative bacteria, another is gram positive bacteria. So, if we have to collect the gram negative bacteria, then we will have to use the medium of which is suitable or which is optimum for the collection of gram negative bacteria, and then the gram negative bacteria is collected. If we will have to collect the gram positive bacteria, for that we will have to use a medium which is compatible for the collection as well as growth of gram positive bacteria. And similarly, in case of fungi, we will have to use a different medium what I have earlier showed in this lecture.
Definitely, as in previous lecture also we discussed about the various types of bio aerosols. Are there any some unique or different kind of bio aerosols?
There are many kinds of bio aerosols. I already discussed that generally as far as these categories are concerned, they are broadly divided into two categories. One is viable, another is non-viable. So, viable is that particular type of bio aerosols which have the capacity to grow, or once it is in contact with a optimum media for suitable for its growth, there it starts multiplying. So, that is known as viable, while another class is non-viable bio aerosols. So, non-viable bio aerosols are those bio aerosols which cannot grow, cannot overgrow, or do not have the capacity to multiply. So, such bio aerosols are basically either generally they are dead bio aerosols like bacteria, fungi which are dead, or they are the part of living organisms such as tree, leaves, etc. If they are eroded or if they are taken away from there, and if they become the part they become airborne, so they come into the category of non-viable bio aerosols.
That was the condition with the bio aerosols, that is when we discussed about the various type of bio aerosols. This is what you explained in the previous lecture also and give justification today also. Bio aerosol. Bio means something living.
Biogenic,
and
yes.
Aerosols means made up of
living
several
is in fact this could be in itself as total biogenic thing, non-living or living, or it can be upon the aerosols which have the chemical nature. I mean, it could be layered or deposited on aerosols having some chemistry. So, if the chemistry is suitable for its growth or for its survival, that particular, let it be, let us take an example of bacteria. If there is a bacteria which is there on some dust particle, and the dust particle is, if it is having, it is moist, I mean if it is a moist dust particle, so the bacteria will have continuous access to water or moisture. So, its longevity increases. So, once that particular dust particle is airborne which is containing bacteria, so what will happen, and because of its moisture content it will survive, it will continue to survive, and once it is inhaled, so from that dust particle now it can go inside someone's or human's body. Suppose the human is susceptible or that particular bacteria is pathogenic. So, once it is entered into the human body, there it might have the capacity to overgrow or multiply, and once the multiplication is full-fledged, then the infection or the person starts suffering from that particular infectious disease.
As you yourself mentioned that these bacteria or the viruses, they multiply. Can we say that in the presence of the all the chemical substances which forms aerosols, these bacteria and or the viruses or something like that, they get appropriate conditions for growth?
Yes. Suppose it has some organic medium. I mean like potato. If there is a fraction of potato which is airborne, and although that fraction of potato is very small, it is of some micron size, having the capacity to be airborne or to be suspended into the air. Once it is been deposited or from where it is originated, it was incorporated with some bacteria. Okay. So, this potato gives a optimum medium for its growth as well as for its survival. So, it is might possible that, and if provided the temperature condition is optimum. If there is an optimum temperature, so the bacteria which is deposited onto that potatoes, a smaller fraction will survive, as well as with its full capacity it will start overgrowing or multiplying. So, initially there could have been 1 or 2 bacteria. Now, with the help of that suitable medium, the number of bacteria increases to many thousands or many hundreds. So, and afterwards if it is inhaled by some human being, once it is gone inside, it founds even the more suitable medium, and there from thousands or hundreds, and it can be in the numbers like millions or hundreds of millions, and then the pathogenicity or the disease to that person starts.
That is the condition which you explained, that how the living beings which in contact with the aerosols, that is the chemical compounds, grow further. And as you said, and you talked about also, if these bioaerosols comes in contact with the human beings, it causes various kinds of diseases to human beings also. So, what kind of diseases one could predict?
In fact, I showed you one picture in the last, that was sometimes back. It was a kind of epidemic, that was SARS. SARS stands for severe acute respiratory syndrome, in which, and in fact SARS is a very dangerous disease, and sometimes back more or less a good portion of world was suffering with the help by this disease. And we take a very recent example. There was an outbreak of Ebola virus in South Africa, especially in Liberia, etc. You would have heard of this. So, these are the, in fact they are actually the viruses, but what happens, the viruses are basically supposed to live or supposed to continue at that place where it is originated. But suppose if the viruses are spread in a dust medium where there is a sufficient amount of moisture with the dust or tree leaves, etc., etc. So, what will happen, those dust or tree leaves, etc., which contains this virus, with the help of air, it can migrate from one place to other place. And there, what you asked just now, that if that carrying medium is having some suitable kind of media for its growth as well as for its survival. So, if it is being transported in few hours or few days, but having a media suitable for its growth, it will survive or it will continue to live. So, once it was generated somewhere else, but now it is been transferred to somewhere, so to some other place, and there if it is inhaled by some person, then it will go into his body, and there that person will start suffering, and now this disease is being spread from one place to other place, and there now the new person which is been who have been infected by this particular disease will start acting as a potential source, and there he will start spreading this disease. Thus, the bioaerosols are basically not pathogenic in itself, but they are also the carrier of pathogenicity.
That is all you explained us about the bioaerosols, and hope that all the students who might have watched us right now would be benefited from this particular lecture. And I would like to tell you all that if you want to access this particular lecture, you can access it anytime with the help of the YouTube, because daily we upload our lectures on the YouTube. Thank you, sir. Thank you so very much. And we would be again with Dr. Arun Srivastava in near future. Thank you, sir. Thank you so very much.